Okay, hi everyone. My name is Miriam Poley. I am first year PhD student in computational biology and biophysics at the Barcelona Supercomputing Center in Spain. I'm here today to talk about our ongoing project, which is called Exploring the Biophysical Boundaries of Protein Families with Deep Learning Methods. We started this project because we want to understand why natural protein sequences are as they are nowadays and how deep learning models could help to understand this regard for evolution. These models are basically algorithms that allow us the design and generation of protein sequences by learning patterns and features present in real proteins in order to do related predictions when inferring. These kind of models in the last years have been classified in so many ways, but we focus on models for sequence generation and also models for reverse folding. This thing of reverse folding, like models as protein NPNN or PROST5, is about using 3D structures as input to generate protein sequences that fall into the given structure. That means generating protein sequences with a similar folding. Um, but models for sequence generation need to learn the grammar of protein sequences and basically how to generate protein sequences in a specific context. For that reason, most of the time, we need the fine tuning to specify the context that we want to explore. But all these models have something in common, and is that from a biological point of view, there is a lack of interpretability. We need to understand and interpret what these models are actually generating. So for that reason, in this work, we propose to measure the energetic of artificial and native ensembles to compare the energetic, but how? Using local energetic frustration. This concept comes from the fact that proteins are not only optimized for folding and instability, proteins are also evolutionarily selected to function. And this could explain that almost the 10% of the internal interaction in proteins are in conflict with their local structure. And this conflict seems to be preserved over evolutionary and physiological timescales, making possible protein functions. The good thing is that we have a tool called frustratometer to measure local energetic frustration. The frustratometer measure how well optimized for folding the energy of a given residue or interaction is in comparison to random variants that we can sample in non-native conformation. So the tool allows to classify all the residues in a structure in highly frustrated, minimally frustrated, and neutral, the three different frustration states. Being the highly frustrated residues, the less favorable to the energetics and related to functionality, while the minimally frustrated are the more favorable for the energetics and related to stability. But it's difficult statistically to interpret frustration result on a single protein structure. For that reason, we are also developing the Frustraivo to measure the conservation of the local energetic frustration. In the same way, we can plot frustration result from a set of related structure to a multiple sequence alignment to have what we call a multiple sequence frustration alignment, and then measure how conserved each frustration state and frustration state and sequence identity, is within each position and the multiple sequence alignment. To summarize all this data, we use this kind of logos where the age of the letters and where the size of the letter and the age of the bars are related to conservation. The bigger, the more conserved. And we can define in any protein family 4 different scenarios. When frustration and identity are conserved, when just frustration or identity is conserved, only one, and when any of them is conserved. And this is basically what we have been doing in native protein families like alpha globins, a good example of protein-protein interaction, and beta-lactamases, a good example of an enzymatic protein family. And we found 3 and 12 highly frustrated and conserved positions in each family. In the case of globin, most of them are related to protein-protein interactions, as we expected. And in the case of beta-lactamases, the three are annotated catalytic site in the literature. So the question now is if the artificial models are able to reproduce these frustration patterns and how similar they are compared to native sequences. So if we generate protein sequences using models like ProGPT2, protein NPNN, and PROST5, we also predict the structure of these sequences. We can apply our frustration analysis to compare the energetic of native ensembles and synthetic ensembles. The problem here is that with these logos and now with so many datasets, it's not that easy to look for patterns. So now, in such cases, we are using consensus sequences, where again, the size of the letter refers to the sequence identity conservation, but only the position with a conserved frustration state is color, but the four different scenarios are the same as before. Let's start with the result with alpha globin from ProGPT2. Here in the logos, we have selected the 12 highly frustrated and conserved functional site, and also just below took the full comparison with native one. As maybe you notice, the amino acid distribution is very close to the native one, and also the frustration state are very similar in between. In fact, all the functional sites are fully recovered. However, ProGPT2 in ProGPT2 ensembles appears new highly frustrated and conserved position. And almost the same situation is what we noticed in PROST5, even when we didn't fine-tuning or retraining the model at all. So for now, we don't know exactly what functional implication these changes proposed by generative models could maybe have, but what is especially surprising is what happened with models like protein NPNN, because protein NPNN is not a generative model, it's a graph neural network, and is trained with structural data. But in this case, protein NPNN is not able to recover any of the functional site annotated in alpha globin. And even in some cases, the functional sites are substituted by less frustrated amino acids, like, for example, the case of the glutamic 27 or the lysine 29. We think that this is maybe a consequence of the sampling temperature, because according to protein NPNN, the amino acid bias is a function of the sampling temperature, and we are generating sequences at temperature 1. For that reason, we are obtaining ensembles, protein NPNN ensembles, more stable than the native ones. But if we check these positions in all the temperatures, in any case, the functional site are recovered, maximizing stability in all the cases. But surprisingly, when we analyze beta-lactamases, the results become more interesting, because in this case, the model, protein NPNN, is able to recover the majority of the highly frustrated catalytic site, even when they are unfavorable protein energetics, because they are highly frustrated. In fact, in this case, checking again all the temperatures, the functional site, the catalytic site are recovered for all the temperatures, and now the amino acid bias and the highly frustrated state remain. So we think that this suppose that this model have a strong bias toward the native identity of functional modules, but more specifically, so for the identity of functional modules, because now protein NPNN ensembles are more similar to the native one. So our hypothesis here is that this is maybe a consequence of the internal intrinsic evolutionary information that the model contains in the training dataset. So to try to prove it, we are mutating in silico all the catalytic site to a different amino acid involving a minimum state of frustration, for example, to valines. We check with FrustraIvo that these valines are minimally frustrated compared to the native identities, and we decide to use the mutants as target to protein MPNN instead of the native target. But we still obtain the highly frustrated and conserved catalytic site. So what's happened? This is consistent with our hypothesis. So what happened if we retrain the model with non-enzymatic data? This is what I did, and this is supposed to remove from the original dataset almost the 50% of the data. And in this case, we have a very, very preliminary result suggesting that at least some identities of the catalytic sites are recovered, which is not conclusive, but at the same time make us think that maybe there is a structural predefinition of the functional site in proteins. So this is something that we need to keep investigating for the community. But for now, we can conclude that artificial protein design is consistent with the native energetic frustration, even when these models have not been trained with frustration data yet. Also, that deep learning-based protein models are really useful, revealing inherent patterns in the evolutionary history of proteins, but especially reverse folding algorithms, because they allow us to sample how much variability the same fold can present, but still recovering functional residues. And finally, that for sure, combining this algorithm with sample interpretation tools could be a powerful way to discover and annotate unknown functional residues. So to finish, I would like to say thank you to my team and also to my collaborators, and thank you to you for coming and listening and open to questions.
Okay, thank you, Miriam, for that amazing talk. We have time for one quick question. If anyone wants to ask. Over there. Let me get you a mic. Just speak up.
I can hear you. I can hear you.
So,
Don't worry.
For some sites, the amino acid itself would be conserved across the different designs, and for some, maybe the frustration level will be the same, but there will be different amino acids. So, so did you look at which happens when, and that may tell you that the bias in that position, or the conservation at that position is for preserving the frustrability, because it's important at that position.
You mean the conservation of the frustration state at individual positions?
Sorry, maybe, so for the same position, sometimes if it's the frustrability is the same, but of course maybe different algorithms or compared to native.
Yeah, for example, in the first two models that I explained, in most of the cases, because we have tried these two protein families, but also more heterogeneous protein families, like for example RAS. In such cases, it's always the same frustration state, but we think that it's because we fine-tune the models. So most of the time, the sequence identity is the same. So when you calculate the frustration, you are calculating the frustration over the same amino acid. So it makes sense if the frustration state is the same. But the idea behind the protein design is to make more stable proteins, but at the same time also functional. So we are trying to find like the equilibrium, because this concept of frustration is from the biophysics of proteins. So in terms of evolution, is needed for the protein. I don't know if this answer your
I
question.
think so, but I think if I read the paper, I'll be able to understand better and ask a better question.
I mean, I have a poster, so maybe we can discuss also later.